My name is Katrina Deckinger. I'm a junior at Teal College, majoring in biochemistry with a minor in medical biology. The discovery of Aquaman was a research project completed in my microbiology course, which is partnered with the CPHAS program. The program is a joint program between the University of Pittsburgh and the Howard Hughes Medical Institute. The program is designed to provide undergraduate students research experience through the study of bacterial phages and their diversity. A bacterial phage, also known as a phage, is a virus that infects bacteria. Through this project, I was able to discover and name my own bacterial phage. The reason for doing this research, its importance, is that phages can be an alternative to antibiotics. It's called phage therapy, and it's very important because of all of the antibiotic-resistant bacteria that are found. When we can't treat bacterial infections with antibiotics, the hope is to be able to treat them with phages. Figure 1 is a diagram that represents all the steps conducted throughout the experiment, and I will zoom in on it as needed throughout the presentation. Figure 2 represents the parts of a phage. The two main parts of a phage are the head, which contains its genes or DNA, and the tail, which allows it to attach to bacterial host. There are two different types of phages, lytic and lysogenic. Lytic phages follow the lytic cycle shown here in figure 3. The cycle starts with attachment, where a phage attaches to a bacterial host. Penetration, where phage genes are inserted into the bacteria. Biosynthesis or synthesis, which phage genomes replicate and then make new head and tail proteins. Assembly is when the phage genome is placed into the heads and the tails are attached. Release is when the new phages exit the cell, killing the host bacteria. In figure 4, the lysogenic phages are different than the lytic phages because the phage genome inserts its genes into the host genome, creating a prophage, which is shown here. The host will continue to divide with the phage genome within it. Upon induction, the phage enters back into the lytic cycle and begins with synthesis going through release. The first step of this project was to collect 2 soil samples from Teal College's campus. We followed the protocol in the CPHAS guide, and our host bacteria was Microbacterium foliarum, or M. foliarum. M. foliarum is a good host because it lives in soil, is safe to use, and easy to grow in the lab. Direct isolation involves adding the isolated phages directly to the pica plate with bacterial host and seeing if any plaques or clearings form. In figure 5, there are plaques which indicate sample 1 was positive for phage. I also did an enriched sample, and sample 1 was also positive for that which is expected. I then picked a plaque and did a sterile dilution. I then did the purification, step 2, more times by picking a plaque and doing a sterile dilution again. On the last time that I did that, I by chance got a web plate. The web plate is shown here in figure 6. I collected a phage lysate from the web plate to figure out the titer or concentration of phage. Additional web plates were created using the phage lysate. Figure 7 is one of the plates made from the combined titer. I got a 6.4275 times 10 to the 9th plaque forming units, or PFU, per milliliter titer, which is a high enough titer to continue on with the rest of the steps of the experiment. I then used the phage lysate to extract the DNA. My lysate was also sent to Pitt to be stored in the phage freezer. Aquaman was observed to be a 24-hour lytic phage due to plaque formation in 24 hours, and there were no halos that formed on the plaque that would indicate a lysogenic phage, which you can see here in figure 7. Further research on Aquaman would be to sequence its genome. I enjoyed doing this research and contributing to the phage collection to help treat bacterial infections in the future. I'd like to thank Dr. Sarah Swerdlow for mentoring me throughout the project and Teal College's biology department for their support.